Hi, my name's Nick. So we're going to be covering a lot today. So I'm going to jump right into it. So first we're going to cover the ear. If I sound a little funny, it's because I have a cut on my tongue. So sorry about that. But we're just going to charge right through here. So that marker does not work. We'll use red. So there's three parts of the ear. We have the outer ear, middle ear, inner ear, okay? So the outer ear is mainly the auricle, which is like the part you can see here, and the ear canal, which leads us to our stirrups or to the middle ear. And the primary job of the middle ear is to transfer air vibrations into what's the right way to say it? Transfer air vibrations into liquid vibrations, essentially, right? Because right here, this is full of lymph, right? Well, there's perilymph or endolymph, and this will be 100, if not thousands of times denser than regular air. So obviously, air vibrations are going to work as well in the inner ear. So the purpose of the malleus, incus, and stapes, or the middle ear, is to, when it hits that air, it will hit the oval window, and that mechanical force will transfer, oh, pardon, my tongue is killing me, transfer the energy and vibrations into the liquid inside of this ear, of the inner ear, right? And that's a very basic overview. And so now I'm going to jump into what this really is, the cochlea. The cochlea is a masterwork of human hearing. Without it, we would not be able to hear. Nevertheless, we'll just jump right into it. So this right here, this view, is like if I unrolled it and like cut it out, folded it, put it out for us to see here. You'll see it makes sort of like a point shape, and you'll have the scala vestibuli, which is on the top here, the scala tympani, and at the top here you'll have something called the helicotrauma. I think I'm saying that wrong, but my tongue's on fire, so we're going to run with it. So the scala vestibuli hits the helicotrauma, comes back, becomes a scala tympani. And you'll see on the inside here of the cochlear duct that there is this blue line. This blue line is meant to represent the basilar membrane. Basilar membrane, super cool. So the neat thing about it is that it vibrates. And it vibrates at a very specific frequency. So usually they like to draw a diagram like this. So imagine different parts of the basilar membrane, sort of like a piano or a violin. I like to think pianos because they're the coolest instrument and everything else smells. And that's a scientific fact. And you'll notice that near the round window here, this top part, there will be shorter, right? The basilar membrane will be shorter and tighter. And then as it goes out and out and out and here, up until the helicotrauma, will get longer and longer and longer. And what do we know about strings? The longer and thicker they get, they only resonate or shake with that which is lower in frequency. And so this is how we determine the pitch of a voice, right? When I speak low like this, you're using the basilar membrane closer to the tip of the helicotrauma, or this endpoint right here. But when I speak like this, like a little fairy boy, it'll be this that shakes. It'll be closer to the round window, proximal to here. And that's a very basic overview. And so now we're going to get to the more chemical side of it. I'm sorry, how well can you see it? Here, I'll get it closer here. Sorry, these markers are monkey water, so we'll just have to work with this. So the endolymph, this right here is full of potassium. And your hearing cells are going to be sitting on top of the vascular membrane, which will shake when required, right? And so what happens is so you have 3 outer hair cells and 1 inner ear cell, right? And so this inner ear cell, or not inner, my apologies, the outer hair cells, what happens is they have these latches here, right? These latches will be connected to other parts of itself, right? Here, let me just check something real quick. And so as the membrane shakes, yeah, I'm right. Okay, I was just double checking. As the membrane shakes, those get pulled open, right? And potassium will rush into it because it'll be shaking up and down, and parts of it is still stuck in the tectonic membrane, that green bridge I have right there. It'll be forcibly opened up. It's sort of mechanical. Think of a mechanical gate, really. The potassium will rush in, and it'll give it a positive charge. And so this positive charge is super cool because on the sides of these cells, you have these proteins called Preston, who is not only a kid in the school I taught, but also a special protein on the sides of these cells. Preston has this neat thing of when he gets charged or stimulated, he shrinks, right? So as Preston shrinks, remember these 3 outer hair cells are still connected to the tectonic membrane. So what's that going to do? It's going to bring the tectonic membrane closer to the inner hearing cell. And here, let me just check this real quick. Yeah, and because the inner hearing cell is also mechanically gated, will get positively charged, like calcium, not calcium, my apologies. Potassium will also rush into it. The tectonic membrane draws closer to it. Will get positively charged. It'll open up its DHP. And it'll be able to release its neurotransmitter. We don't know for certain what it is yet. A few scientists have postulated that it's glutamate, but reality is we don't know yet. And so that's a super brief overview of hearing. There's 3 kinds of hearing loss. There's central hearing loss, sensorineural hearing loss, and conductive hearing loss. Central hearing loss is your brain is just cooked, right? You had a stroke and that part of your brain either was damaged or died. And there's not a lot to come back from with that. Sensorineural hearing loss is when something messed up in the cochlea. Like, let's say you call it cytomegalovirus, and it damaged your basilar membrane. Actually, we don't know the pathology behind cytomegalovirus. I'm just a big fan of that. I studied that way back when. And then there's conductive hearing loss, which is the sound just going to make it through, right? Let's say you had a cotton ball in your ear, or you have so much wax in your ear, it just didn't work out. That's a very brief and simple overview of hearing. It's pretty complex. I think it's super cool. I'm going to check to see if I missed anything. Ooh, this is super cool. So you know how you're able to tune out voices? So your brain has neurons connecting to this right here. And it's able to sort of counteract the Preston, either by hyperpolarizing it or whatever, and to make it to stop stimulating to certain voices and stuff like that. So like, let's say you're in a crowded room talking to that cute girl in the party and you only want to talk with her, your brain's going to help you out and shut down these hearing cells for all the other sounds, because that's not important. Until, say, you hear your name, your brain picks that up, and the subconscious is like, oh, hold up, we got to pay attention to this. It'll stop interfering, no longer hyperpolarized. It starts to open up again. That's really cool. You have the stria vascularis, which is, primarily in charge of keeping the endolymph full of potassium and the paralymph not full of potassium. That would be quite bad. Okay, I'm just making sure I didn't miss anything. I think we got that pretty quickly. All right. Let's move on. Okay. So I think next we'll cover smelling and taste. Sorry, just have to check, make sure that was working. Okay. All right. First we'll cover smell. Okay, well, I'm trying to think of how to best draw this. That is atrocious. I am so sorry. Can you even see that? Yes, you can. It'll work though. Is that good? I think so. Okay. So we have the cribriform plate right here. Special part of your skull lets the neurons that connects to your brain for smell go. I think it's pretty cool. Fun fact: your olfactory bulb and like your olfactory nerves themselves can reheal. But let's say you get in a car accident, and your cribriform plate shifts and cuts off the axons. You're not gonna have a sense of smell anymore, which is all right, I guess. But hey, wouldn't want to be me. So we're gonna cover real quick of how this works. So here's a closer look of your olfactory nerve right here. Give him a nice Squidward face. Drawing that beloved G protein. All right, and then here's our channel. All right, I think that's about good. Yep, okay. So essentially what happens is G protein gets activated, sends out C-camp, goes to here, opens this up, calcium, well, what really happens, not really calcium, sodium, once this channel is opened by the C-amp, sodium will rush in, create a nice wave, and that will activate your neuron and send a boom to innervate them. I'm pretty sure that's it. Let me just check. Yeah, that's about it. Yeah. By the ways, COVID enters through ACE2, which is a part of your nose here. I think that's pretty cool. Oh, that was pretty interesting in case you're interested in that. That's why COVID can destroy your sense of smell. Because it actively attacks that, or enters through. I'm not a COVID pathologist, but that was my understanding of it. All right, so taste, sorry. We'll start with the, what am I thinking of here? Sweet, bitter, and umami. Then we have salty and sour. First, we'll start with salty. It's very simple. So on your tongue, you have these chemoreceptors. Papillae. Your papillae, papillae. And so they work like this. right? And in this case, for salty, it's reactive to sodium, as you could probably predict. There's a channel for it. And on it, there's a DHP. When it gets positive enough, it'll release glutamate, right? To the neurons underneath to innervate it and tell your brain, like, hey, this is happening, and that this food is salty. Next, we have sour, which is very similar, except the channel is for protons. You also have a DHP for this, which is just your calcium, right? And the calcium activates your vesicles for glutamate, right? And then it runs through, does the same thing, right? Same stuff, different pile. Next, you have sweet, salty, sour, sweet, bitter, umami. Yeah. Umami is like Japanese for flavorful. It's pretty cool, I guess. Like, I'm just goofing off. So you have a G protein here, right? And you have your subunits. X receptor goes off, triggers it. Alpha will go to, I think it's PDE. Is it phosphodiesterase? Or am I confusing that with vision? Or is it PLC? I think it's PLC. It's a PLC. And now the signal becomes IP3. Hits the sarcoplasmic reticulum. That should be familiar from the skeletal system because there's a calcium pocket here. The calcium pocket will then activate the vesicles inside and then tell the neuron outside of it. These three are unique because it gets its calcium not from outside but from inside. And so I think that's pretty notable and interesting. Let me check to make sure I didn't miss anything. I think that's about it. There's also spice, which comes from capsaicin, which is the reason you taste salt occasionally. Not salt, spice, right? That's why the Korean food will like destroy you. All right. That's it for taste. I just mainly wanted to cover the senses and how we taste different things. I think that's really interesting personally. Alright, so I got smell, taste, hearing, eyes, and ligament. So now we're going to talk about the eye, which I find to be really important. And you'll see why in a sec. I'm not going to apologize for that. That was funny. Ooh, I did a good job. Here's your... All right. So I'll make sure I'm getting this drawing right here. I don't lie to you guys. Yeah, that's the iris. Okay, just triple checking. You have your iris, you have your suspensory ligament. And then you have ciliary muscle. Although for what I'm about to talk about, maybe you'll want to call it the ciliary sphincter. Trust me, that'll be useful in a sec. And so, remember, when this muscle contracts, the ciliary muscle, it only closes. And so we have the lens here. And the job of the lens is to bend light so it can focus on your retina. I almost misspelled retina. That would have been embarrassing. Retina works like this, or lens. So let's say my hands here are the lens, all right? So when the ciliary muscle contracts, it comes closer to the iris, and these suspensory ligaments will loosen and go and lax, causing my lens to fatten and widen. And as a result, it will be able to bend light better, which is important because you need to bend light more if something's closer to you versus farther away. It goes like this, right? It's like this, it goes like this. Versus, say, farther away. Where it can be a little less aggressive. But it does need to relax. Versus when something's farther away, the ciliary muscle will relax and widen up, tautening the suspensory ligaments, pulling the lens to be straighter and not refract as much. Because if it refracts too much when it's not, you're going to get blurry vision. And that's a very basic overview of the eye lens and ligament and how they behave in relationship with each other. And now we're going to talk about these guys right here. The guys picking up the signal next to the epithelial cells that absorb extra light, your rods and cones, your photoreceptors. And they are the superstars. Well, it's a team effort, but they do a lot of the heavy lifting. So, all right, let's get a photoreceptor up here. We've got our opsin. Well, I should call it rhodopsin right now. Your P, D, E. And up here in the corner, you have your, what's it called? Don't tell me yet. Yes. It's going to take me a sec, but you'll appreciate me once I get this all written out. All right, got this. And ganglionic cell, which goes straight to the brain. All right, I don't think I missed anything. Yeah, no, I got it. I'm going to call this an on center because that's what it is, an on center, and I'll talk about what that really means later. But this is a photoreceptor. You have your photoreceptor, bipolar cell, or neuron, and just a ganglionic cell. And so, all right. First we start at the opsin, this G protein here, or rhodopsin, since retinol is still in it. Retinol will cause this cis bond to straighten. Once it straightens, it'll leave and activate this messenger. The messenger goes to PDE, which activates the photodiesterase. The photodiesterase then in return destroys this CGIMP, or CGMP, which will then close this sodium channel you see here. Really important, because this stops it from polarizing, or depolarizing, I should say, anymore. And this DHP will begin to close since it no longer hits the voltage requirement, which will make it so glutamate, which is being produced by this, or released, I should say, not produced. That's I imagine, a complex process. I don't know where glutamate is produced. Being released from this photoreceptor. And so it's sort of counterintuitive, but you need a light to tell your on centers to stop producing glutamate. And that's how a message begins to get sent. So by not doing anything, that's when a message is finally sent. Because this GLUR6, this I think it's a G protein, this G protein here, once it stops getting hit with glutamate, it's like, oh, okay, now I'm allowed to open this TPRM channel right here, which will open this up and enable, wait, let me check something. Yes, okay, I'm right. I forgot to put down a DHP here. So sodium will rush in, depolarize the cell, and it will cause these vesicles here, also filled with glutamate, to finally release. But in this case, we want the glutamate to release because in that case, it will innervate this ganglionic cell, which will tell the brain, hey, we got light going on here. And we know that because we're finally getting innervated. Because if glutamate was able to still transport here, the GLUR6 would keep this channel closed and we wouldn't be getting a message from the bipolar neuron. You might be thinking, like, okay, if that's an on center, what's an off center? Surely it does something different for the photoreceptor cell, and the answer is no. It actually changes the bipolar cell. The bipolar cell, instead of being a GLUR6, it becomes an AMPA. And AMPA actually wants dark because when it gets hit with glutamate from the photoreceptor, it opens up, and then, you know, like sodium can just run in that way. It wouldn't really need this TPRM. And it would then release signals to the brain there. And so now that we got a basic understanding of that, we get to cover the last of the stuff, which is to say center and surround signals. Photoreceptors don't work alone. Photoreceptors need support. And just like any movie star to shine on the podium of life needs help, so too does your photoreceptors. So, like, here's an example. Here's your center. Oh my gosh, that was atrocious. I'm so sorry. And here's your surround. And they're always opposites, your centers and surrounds. And oh yeah, they'll be adjacent horizontal cells. And so if the center is on, then these are off. And we'll act as though that's the case. So this is how your surround cells work. One thing to remember is that glutamate or light always reduces glutamate, right? And so let's say light hits the entirety of this region of the eye, right? You have your photoreceptor, it has its two surround cells. And so light's hitting all of it. And so the primary job of the surround cell, assuming it's like working in the dark, is to activate these horizontal cells with glutamate, which in turn releases GABA, a neurotransmitter that inhibits the center from releasing more glutamate, right? And so if these release glutamate, it will inhibit the center from releasing glutamate. If these don't make glutamate, it won't inhibit the center from making glutamate. And so remember, This is all leading up to the same bipolar cell. And since the center is on, we know it's a GLUR6. You got it perfect. All right. And yeah. And so center on, we already talked about this. The light will make it produce less glutamate. But let's look at the surrounds now. All right. And so these are supposed to be off, right? And so light hits it, it'll produce less glutamate, which in turn makes this horizontal cell less able to produce GABA, which in turn makes it less able to inhibit the center cell from making glutamate. So percentage-wise, we usually call this 6 and this 2 and 2. If you want to go all in on the percentages, you can say 60%, 20%, 20%. So if you have light on the whole eye, how much of the glutamate will still get released? It would be 4 or 40%. Glutamate would still get released. And so it's a really smart system you got going on with this center and surround, because your eyes are able to get all these different clusters and such like that. And from the number of impulses the brain receives from that region, they can tell how dark or how bright that region is and what part of the receptors are hitting light. Like let's say light is only hitting these two guys. Well, in that case, this guy is still able to make a ton of glutamate, makes a ton of glutamate, releases it to a horizontal cell, which in turn makes a ton of GABA, which is able to hold on to that. Wait. I'm sorry, I mixed it up before. When light was hitting all of it, it would release 6 impulses because the light has been properly inhibited from releasing 6 of its glutamates, which is good for the GLUR6. But returning to my original point, I apologize. Let's say the light is only hitting these two. Like this is inhibited from producing that glutamate that would inhibit more glutamate from being made. But this is in the dark. So a ton of glutamate made, more GABA is made, which inhibits 2 more glutamate from being made. Now you have 8 impulses or dashes from those receptors from being released. And that's really important. And if you're wondering what would happen if the center was off, then your surrounds just turn on and you flip what I said on its head. And that would be it. Yeah, thank you for watching and have a great day.